This is the third presentation in the series about the biological basis of learning disorders. This presentation looks at dyslexia. Dyslexia is a disorder associated with accurate word recognition, poor spelling, and slow decoding abilities, each of which affects the ability to read and to write. Dyslexia is surprisingly common, affecting about 18% of school children. It's important to recognise that dyslexia is a spectrum condition, broadly categorised by reading difficulties. About a quarter of children with dyslexia have severe reading difficulties. There is no single cause of dyslexia, although dyslexia is known to be associated with brain development. Dyslexia is not associated with general intelligence or IQ, nor is it associated with severe sensory deficits, such as visual impairment. It is perhaps not surprising that there is no single cause of dyslexia. Learning to read is a complex process involving many aspects of vision, language, the motor control of eye movement and attention, and it's interaction of all of these processes that lead to the ability to read. Individuals with dyslexia are also frequently observed to have impairments recognizing auditory and visual stimuli in the presence of background noise. It looks as if dyslexia is the reduced ability to distinguish intentional stimuli from perceptual background noise. However, two parts of the brain do seem to be particularly crucial in coordinating the various processes involved with reading. These are known as the visual word form area, VWFA, and the left intraparietal sulcus, IPS. We might expect genetic factors that influence dyslexia to be operating in these regions. Brain imaging suggests that there are differences between a dyslexic group and a control group in their responses to hearing a person's voice for a second time. The control group shows a stronger ability to recognise the voice for the second time than the dyslexic group. This does suggest that there are functional differences in the brains of dyslexic and non-dyslexic people. An important new paper by Gyrus et al. In October 2020 is a genomic study of dyslexia. They suggest that developmental dyslexia has a heritability of between 40 and 60%. That is, up to 60% of the differences between participants in the study are caused by genetic factors. But there is no single gene that causes dyslexia. Even so, they have reported some protein coding genes that are significantly associated with dyslexia in their study. Some of them, such as a gene on chromosome 20, has not been characterized before, whereas other genes, such as the one on chromosome 3, are already known to be implicated in brain development. Of particular interest is that the genetic factors that they highlight seem to be active in one particular part of the brain, known as the transverse temporal gyrus, where there was a positive correlation of cortical thickness with a reduced risk of developmental dyslexia. We know that cortical thickness improves the ability of the brain to transfer and process information. So a thicker cortical would be expected to lead to improved information processing. The association of many of these genetic factors with this area is significant, not least because that particular part of the brain is known to process what's known as the inner voice and is already known to be implicated in dyslexia. One interesting possibility arising from this paper is the possibility that some non-coding genetic variants, SNPs, contribute to more than one characteristic. For example, some of the SNPs which are known to be significant in this study of dyslexia are also known to be contributing to characteristics such as attention deficit hyperactivity disorder, schizophrenia, cross-disorder susceptibility, educational attainment, and general intelligence. This suggests that these variants may, if they do have a role, will be a general role in the development of the brain. This is a significant paper and does clearly show that there are inherited influences on dyslexia. It was a large study involving 2,274 dyslexia cases and about 6,000 controls from a number of European-speaking populations. In total, the genetic factors identified in the analysis account for less than 25% of the total variance in developmental dyslexia, which is far less than the 40% that was expected from analyses of twin and adoption studies. So about half of the variation is missing, half of the heritability is yet to be explained. This is a current area of debate within developmental and genomic genetics. It's possible that it's accounted for by variations that are so rare that the analysis did not pick them up. It's possible that it's accounted for by factors which are duplicated across the genome, so-called copy number variants. But it's possible that the twin studies and the adoption studies estimates of heritability are themselves subject to error and bias. At the moment we don't know and cannot explain why the heritabilities are different.